I'm really excited to introduce our first speaker. She has an extensive background in science, from her degree in college to past work experience, and she's actually spent 10 years with Sony Biotech, and she is here today to tell us a little bit about her job and her career. So Carla, welcome. We're excited to have you here.
Hi, thank you so much. I'm very excited to be here. Hi, everyone. My name is Carla, as Nicole said. I just wanted to start with my background. Go ahead to the next slide. So I started at Portland State University. I have a Bachelor's of Science degree in microbiology. That's in Oregon, all the way in Oregon. So after my degree at Portland State, I got into the food industry. I was a microbiologist, and that means that I tested food to make sure it's safe for you and your family to eat so you don't get sick. One of my jobs I had after college was at a cookie factory, so that was super fun. Part of my job was to test cookies, so I would go and eat them. What's your favorite cookie, do you think? Mine was always chocolate chip, pretty basic. Next slide. So after I worked for a couple years in the food industry doing microbiology, I moved all the way from Oregon out to Virginia because I was ready for an adventure. I was young and just any sort of adventure, I was there. So I moved out with my husband and we started fresh. Next slide. And when I was in Virginia, I was able to get a position at Eastern Virginia Medical School in Norfolk, Virginia, and I became a research assistant, and that is where I was able to learn what flow cytometry was and how to do flow cytometry. So that's the focus of Sony Biotechnology, is flow cytometry. And after that, for about a year or so, I learned about flow, and then I was able to go to work for Sony Biotechnology as a field service engineer. So I was able to learn how to repair instruments for our customers. So I did that for a few years, and then I was able to fine-tune my skills and become the product lead for one of our cell sorters. And I was able to learn new skills and share those with other people, both in North America and worldwide, through training sessions, in addition to just basic repairs in the field. And then after I've done that for a few years, I was promoted most recently in the fall to a field service supervisor, which is my current position. So when you are a field service supervisor, that means that you're in charge of a group of engineers. So you may be wondering, who is Sony Biotechnology? Because all of you are familiar with Sony. Sony makes TVs and robotic dogs and headphones and the PlayStation, very exciting, but they also are in biotechnology. So it all started back in 2010, where they purchased an existing company called Eyesight Mission Technology, which they're located in Champaign, Illinois. So after a few years, they decided, let's rename the company Sony Biotechnology. And then in 2015, they moved from Illinois out to San Jose, California, which is one of the biotech hubs of the country. Next slide. So these two products are flow cytometers that Eyesight Mission Technology originally produced. So Sony, when they purchased Eyesight, they also own these instruments. So the one on the left is a cell sorter that I actually trained on. Next slide. And now, present day, we have 7 instruments that Sony has made, and they look beautiful and they function very well for our customers. But just look at all this. In just a few short years, what we were able to do. And these, a lot of times our customers, after we install them at the sites, they ask us, where do I put my coffee mug? Because they think they look like espresso machines. But I guarantee these are not espresso machines. These are very high precision instruments used in medical research. Next slide. So how much do you think these cost? They actually cost as much as 10 cars or a nice house. They are super expensive pieces of instruments. So that's why it could take years to be trained in order to repair them. Next slide. So you're probably wondering, what exactly is a flow cytometer? Maybe you've never heard of it before. So a flow cytometer is an instrument that research scientists use when they're trying to understand more about the cells that they're studying. So say, for example, you are studying cancer cells. What they would do is they would attach fluorescent markers either to the outside or the inside of the cell, and they would put them in a tube, a test tube like in the picture, and run them through the instrument, through fluids such as salt water or saline, and then lasers would actually intercept the cells, and it would cause a signal to come off the cell, which the computer interprets as data on the computer screen. And in some cases, the scientists may want to go further than that and actually sort the cells into test tubes if they're looking for a specific cell subset, for example, for their research. And what they can also do, if they don't have enough of these cells and they're still living after they've been sorted into test tubes, they can grow more. So instead of, say, 5 cells, you can get 10,000 cells. So that's great too. And so the main subsystems of the instrument, so that means what's inside the instrument working together for the final result, are systems such as fluidics, electronics, and optics, or lasers. So that's what I focus on. Myself and my engineers, we have to have a great understanding of each of those subsystems, how they work independently and together in order to troubleshoot and fix them. And I wonder what your favorite subsystem might be, what might interest you. Next slide. My favorite subsystem would definitely be lasers. Lasers are so cool. What I like about them is that you can align them so they hit the center of the cell. They're colorful and bright. And what happens when the laser hits the cell is like I said earlier, there's a fluorescence that comes off, a signal that comes off the cell. So that's really cool. I like lasers. I hope you do too. Next slide. So what does a cell sorter do? I'll give you an analogy to try to explain it because I know it can be complicated to try to understand. Next slide. So if you were looking through a microscope and you can only see a bit of this crayon in the image, you may not know, are there other crayons there? What colors are they? Are they jumbo size, or are they just regular crayons? So we use a flow cytometer to give us more information about the crayons or cells, such as blood cells, for example. Next slide. So in this picture, after you look at them with a flow cytometer, you can see all of the crayons that we're looking at in detail. So you would see all the components of blood in great detail. Next slide. And then you can also sort out the crayons or the blood cells according to color or particular type, for example. Next slide.
That's awesome.
So the real life usages. So we sell our instruments to people in the pharmaceutical industry and universities, colleges, sometimes small startup companies, but they're all dealing with small organisms and cells you cannot see with your eye. So that could be bacteria and viruses, it could be blood cells, it could be cancer cells, different things like that can be used on our instruments in order to find cures for diseases or treatments for diseases if we can't cure them. So basically our goal is to provide instruments that can help make people feel better. Next slide. And also, since we're in a pandemic, unfortunately, our instruments are being used by several of our customers in order to find treatments and hopefully very soon a vaccine for COVID-19. Because what we're trying to do with using our instrument is to learn more about the virus, because that's why it's so hard to find a vaccine. We just don't know enough about it. So we can use instruments like a flow cytometer to find answers and to find a cure.
Real quick, Carla, before you jump in, I just want to remind everybody to share your questions for Carla in the chat, and we will get to those at the end. So if you have any questions on this product or how it works or things like that, send those questions through in the chat and we will answer them in the Q&A at the end.
All right.
Thanks, Carla.
Awesome. So what is my role like being at Sony? So I work in the service department. We have other departments as well, but I work in the service department. And so when we have an instrument that's already installed at the customer site, the customer may go to use the instrument and it doesn't work. So one of my jobs when they call me for help on the technical support line, I have to walk them through a series of questions like a detective to try to figure out what's wrong with the instrument, what specifically is wrong, which subset of the instrument or subcomponent rather is having the problem, and so we can figure out how to fix it. And there might be it might be something super simple. We can fix over the phone or over a remote session, because we do oftentimes remote in to the customer's computer, and we look at the software. We have special service software that we use. It could be a quick fix, or it could require a service visit. So I'd have to figure out what parts might be needed, and then which of my engineers are trained to do the repair, because we have so many instruments, maybe only specific people are trained, and then we'd have to arrange for a service visit. So there's a lot of detective work that goes into this job for sure. I hope you would like being a detective. If so, this could be a good job for you. Also, when you're talking with a customer, or if I'm assisting an engineer trying to troubleshoot a repair, you have to listen. It's very important to listen to every detail that the customer is telling you, because that will give you a clue as to what component of the instrument is not working, so we can fix it. So a lot of times I will have just a notepad sitting on my desk, and as soon as somebody starts telling me the story of what happened, what was happening before it broke, and then when it broke, I'll just be writing everything down, and then afterwards, I will upload that. I'll have to type on my computer and upload that to our database. So we are definitely reporters in this job. And then the other half of this, besides just when customers have problems with the instruments not working, is we have to train engineers. So how do you fix instruments? So I spend time helping develop training programs, how to improve repair techniques, things like that. And then sometimes I end up being the student, because my engineers will come up with a different way to fix something to become more efficient. So that's great. So I'm also not only a trainer, but a student. And then lastly, but not least, an adventure world traveler. So this job, I've been working from home since the pandemic, but we just started to travel again, me in particular, and you never know where this job will take you. Right now, I've been traveling just in North America, but in the past, my past roles, it can literally be all over the world. Next slide. And these are just a few of the places that I've gone in my past 10 years with Sony. I went to Japan, for example. I did some training there where I was trying to learn how to repair the instruments from the team in Sony directly. And then I've done service visits in Europe when the instruments haven't worked, and in the Middle East. and then also training sessions in Switzerland. And this is very exciting because I grew up in a very small town, and I never thought I would be really traveling anywhere per se, maybe just in the US. So the fact that this career has allowed me to have the opportunity to travel it's very exciting and amazing, and I'm thankful for that. I bet you guys have traveled places maybe before the pandemic, or where would you like to go in the future after the pandemic? Next slide. Okay. What are the skills needed for this job? So being able to troubleshoot. So what does that mean? So if you have a symptom, you have to be able to break that down and then start from one end of where the problem could be and follow stepwise until you get to the end. And 100% of the time, the solution is somewhere from point A to point B. You just have to be very methodical and break down the issue and be a detective and really pay attention to what the customer is saying as far as symptoms go. Why isn't the instrument working? Maybe you like to, take things apart to figure out how do they work. This would be a good job for you then. And you're doing it in relationship to science, because the end product of your work is that even if you've literally taken it apart to fix a problem, it's got to go back together in the end and function for our customers so they can use it for their cells, or for if they're sorting or working on viruses or bacteria. It has to work for them. So it's very rewarding. You do have to have a college degree. A 4-year degree is best, but it is possible to have a 2-year degree. You could maybe study something such as electronics. A 4-year degree, like what I did in science, is very helpful because it allows you to understand what you're doing with relationship to the instrument. So it's not just you fix a pump or a regulator. It's you understand on a broader scale how the instrument works and what the customer is using it for, biologically speaking. It does help. You could also study like mechanical engineering. That would also be a great field. So you can have quite a variety of background for this type of job. And then also being flexible, and that's both mentally flexible and physically flexible. So this is a very manual job where you're repairing things. So you might need to squeeze into a very tight location to unscrew maybe a laser from an assembly, something like that, to realign it. But then also you need to be flexible in that you never know where you may need to go to help a customer fix their instrument. So it can be a little hard to make plans, but we have to put the customer first. It's very important. And then it helps if you're very detail oriented as well. Next slide. Okay. Ooh, and what do I like most about my job? I love working with people. I love working with customers. I love working with we have a department, an applications department, which they work closely with the scientists to try to fine tune their research so they can get the most out of the instrument. Also like working with, I think I already said customers and engineers. I like working with fellow engineers because then I can improve my technique. Because one thing I've learned is that even if you've been with a company for a while, like I said before, we still do not have all the answers, and that's okay. We can all work together as a group to find a solution. And then I enjoy coaching engineers and mentoring them as well. It was hand in hand. And Sony Biotechnology offers a great family work-life balance. So that means even though the job it can be tough and demanding because our customers' research is very critical to them, we still have, of course, holidays off and weekends off that we can spend with our family. Next slide. So this is a picture of my family. I have a son, he's 2. His name is Harrison, and my husband. And then that's a picture of me working at a site, trying to do an installation. So if you work hard, you can have it all. It's true. Next slide. So if you are interested in learning more about flow cytometry or science or engineering, I found a few links that you can go to start learning more. It could be helpful if you're interested in a career like this. Next slide.
And we will share these after, so you guys will have access to all of these links.
Awesome. Okay, so laminar flow, this is a principle of flow cytometry that's used in flow cytometry. So when particles go through a channel filled with fluid, such as this in this image, they don't mix. So the colors do not mix. So this will be one of your challenge activities that I've put together for you guys from the internet. Next slide. That will demonstrate laminar flow. So I would suggest checking out this video first, and this just uses pretty basic materials you could find in your house to demonstrate that colors do not mix. It's really cool, but check the video first because then it would make more sense. In the next few slides, we'll just have the materials and the actual method. Perfect. All right, so in closing, I want you to always ask why and be super, super curious about the world around you. And I look forward to learning your story about your career growth. I hope you enjoyed this, and I hope you love science because I certainly do. Thank you.
Amazing. Thank you so much, Carla. Michiko has been moderating the chat, and I know we have quite a few questions in there, actually. So, Michiko, do you want to share some of those questions that you've heard?
Absolutely. They're really good questions. So, Carla, does the color of the laser matter? If so, why?
Yes, it does matter. So each laser is a different wavelength of light, and that is specific for the fluorescent markers that the scientists add to the cells. So each marker is a different wavelength, so you need a specific laser for that particular marker. So yes, laser color matters. Absolutely. That is true.
Interesting. And many people are asking about COVID-19. Can you explain more about how the instrument help find cures for COVID?
So what we're trying to do with the instrument is we're either looking at the entire virus or just a subset of the virus to find out how it responds to different treatments and if it's successful or not. So a customer might go through different iterations of it could be different treatments, or it could be different amounts of one treatment, and it kind of measures the response of the virus to see is this helpful or not.
Great. Another question. Other than your degree, what life experiences and interests have helped you to do your job?
Let's see. Oh, that's a good question. I've never really thought about that before. So probably what's helped me is being able to, so enjoying traveling has helped, and being flexible and open to change. So for instance, when I moved all the way from Oregon to Virginia, so I'm very interested in new things. So that helps you also with technology, because as Sony Biotech makes new products, then you have to be very interested in the new technology that comes with that, because in science in specific, typically it's not the same. So it's always changing. So you have to be interested in technology and very good at computers. And I was always interested in that. In college, I took a class for developing web pages, for example. So I have interest in tech, I would say, and that helps.
Great. And you also mentioned that you are very curious and ask lots of questions growing up, right?
Yes. Yes, exactly. And I would always say, never feel bad to ask a question, because chances are, if you don't know the answer, someone else doesn't know either. So it's always okay. You should always feel safe. And even if somebody discourages your question, that's not your problem. That's their problem. It's okay to ask questions. Always. We're always learning. Even me, 10 years in, always learning.
That's awesome. Okay. How does the machine detect a virus in the cell?
Well, so the instrument, it wouldn't necessarily detect the virus. So the researchers would be studying the virus. They could be studying the virus separately, or perhaps what has happened to the cell after the virus has entered the cell, if that makes sense. It's kind of a little vague. Sorry.
Okay. Next question. Many people are asking, how does the cell from the instrument move to the computer? What's the transition like?
Yeah, good question. So the cells start in a tube that's set onto the sample loader, and then when the customer presses start on the computer, it actually moves up, and the chamber is pressurized. And so when it's pressurized, it moves from the area of high pressure to low pressure, which is this sorting chip. And I'll show you this right here, which is the sorting chip is in the instrument. So I don't know if you can see this very well. So the sample comes in through this bottom port, and this middle port has the fluid called sheath fluid, which is like salt water basically. And it mixes, and then it comes down here, and then the laser intercepts the droplets which contain the cells, and then the signal that's given off, the fluorescent signal, is detected by the data acquisition system in the instrument. And then that signal is taken from analog, converted to a digital signal that the computer can recognize. And then it shows up on the display as a certain pattern of pixels or dots. And the scientists they understand what that means. So it's useful to them.
Wow, interesting. I'm sure many people said, wow. Awesome. Okay, another interesting question. What role did high school mathematics play in functioning and thriving at your job?
Let's see. I would say what was most useful was statistics, I would say. So I use statistics in my job when there's a situation, say the instrument isn't working right, and we can get the data from the instrument, and then I can analyze it to break it down to figure out if it's really not working right, or if it is working right. And I do a lot of averages, different things like that. So statistics is most useful for me, but I do not use anything. I do not use calculus, nothing like that. But math is good, I would say. The more math you can get is better. But for my particular role, statistics is most useful.
Good. People are asking about your travel and what did you learn throughout your journey when you travel? And I would ask, where is your favorite place?
So what I've learned when I travel, let's see. I would say be patient, because when you're going into other countries, you have to go through customs, and it can change so rapidly, and it's unique from country to country. So I remember when I went into the Middle East, it took probably over an hour to get through customs. So imagine you've just traveled about 13 hours, you're tired, and then you have to collect all of your luggage and stand in a line for a while. So that was, I learned a lot about patience and just being flexible. One time I remember I went to France, to Paris, to do a repair, and I arrived and my luggage wasn't there. And so, sometimes you pack a little extra in your backpack just in case, but it slipped my mind. So that's okay. I went to the customer site in what I traveled in jeans. So you have to be flexible and just do the best you can. So the most favorite place I went to was probably in the Middle East, the United Arab Emirates, Dubai. It was surreal. It's a very modern city. There's a lot of money there. The buildings are all new. At the time, it had one of the tallest buildings in the world, the Burj Khalifa, but it just seems out of this world. I couldn't believe I was there. And the food was amazing, a lot of Middle Eastern food. That was very cool.
Great. Nicole, do we have time for more questions? A few more?
Yeah, we probably have 1 or 2 more questions.
Sounds good. The question is this about DNA?
Oh, that's a good question. So the instrument can be used to look at like chromosomes, for example, and DNA. So you could use the instrument for that. Yes. Most popular though, or most typical is for cancer research. That's super common. But yes, you can use the instrument for that.
Great. So last question, is your job fun?
Oh, is it fun? I would say yes. I am never bored and there's always so much to do to help the customers. Yes, it is fun. Sometimes it's stressful, but definitely fun.
Amazing.
Great.
Well, thank you, Michiko. Thank you, Carla. I hope everybody learned a ton from this.